Всё, да, звук есть.
Hello guys, start collecting to listen to a lecture. Today we have immunology lecture. Guys, start coming here, start collecting here. Guys, do we have anyone there? Or, honey, good afternoon, you are the first one here. Good afternoon, my dear. So, and the rest, please, just Winnie and the others. Okay, now we are going to discuss today immunity to microbes. Good afternoon, everyone. Amisha and all others. So, you see those guys, they are really different, right? So, and we try to discuss how the immunity will be developed against bacteria, viruses, fungi, protozoa organisms, and helminths. Truly, we have discussed some of the features already during this week class, but as far as I understand, so you don't know much about this. So that's why I consider it very useful to listen. So it will be a bit more complicated than we discussed during the classes, but then you take a look at this and you will choose what you can understand and describe during the classes. So first I would draw your attention at the general features of immunity to microorganisms. So it means there are several general features with no regard to immunity development against each particular type. So what are those general features of immunity against microbes? Against any microbe, the defense will be mediated not by separately innate or acquired, but by both innate, you see here, and acquired immunity systems. So it means the innate immunity mechanisms will react first and will respond first, then they in turn will activate the acquired immunity mechanisms. Then the second important feature, this is specialization of acquired immunity, or I would add, by the way, also specialization of even innate immunity. So, and this specialization allows the host to respond to different types of microbes selectively. So, what is it about? You know that, for example, macrophages will be more effective to engulf some larger objects. For example, bacteria, fungi, or even protozoa, right? So while the viruses won't be easily engulfed like this, they most probably will infect those macrophages. So then, and of course, we have to pay attention at the specialization of acquired immunity. So, and that is the ability of the host to respond differently to different types of the microbes. So, for example, as a result of this, we may have the synthesis of different isotypes of antibodies. You know that secretory IgA antibodies will protect our mucous membranes. So, if the microbe coming from the site of entry of the microbe is the epithelium, so then the secretory IgA antibodies will be of utmost importance with no regard to whether it is bacteria or viruses. For viruses, we may give example as the rotavirus, okay? And for bacteria, we can give example as cholera. So then the second demonstration of the specialization. This is the generation of different subsets of effector CD4 cells. We have discussed already that basically 3 to 4 different subpopulations of T cells may be important in development of the immunity. And these are Th1, that develop mainly immunity against intracellular parasites, Th2 and Th17 that may develop immunity against extracellular parasites and very strong inflammatory response. So that was about specialization. Now the next point is that some microbes, both bacteria and viruses, may cause so-called, you can see the word here, latent or persistent infection. And then this is the way how these microbes escape immune mechanisms. Because the immunity in latent or persistent infection usually may control the multiplication and spreading of the microbe, but cannot eliminate the microbe. My dear, sometimes the task for the immunity and the best thing it can do is to control the multiplication. And there is no chance for immunity to eliminate the microbe. So we may give a lot of examples. And the best example is herpes virus, because once it infects the host, so they will stay there forever. And you know a lot of examples from medical biology department about the protozoa, for example, malaria organism, right? So that organism that causes malaria, or helminths, by the way. So they cause persistent infection and the immunity can hardly get rid of them. So then, one more very important general feature of the immunity against microbes. Most probably, the tissue damage will be seen in the host. So, but then the tissue damage is performed or is developed not just because of the microbe, but because of the immune response developed against this microbe. And as you can see here, the immune response sometimes may cause the greater damage than direct damage by the microbe itself. And that is the true, for example, for Mycobacteria tuberculosis infection, where the tissue damage is done both by direct action of Mycobacteria, but mainly by indirect action of the immunity that will be hyper exaggerated by the different components of the Mycobacteria. And so that were the most important. We gave 4. Of course, we may give some more, but that's enough. So now, we take a look at the immunity developed against particular types of bacteria, particular types of microbes or pathogens. And we start with bacteria because different types of bacteria may be located intracellularly or extracellularly. We have a lot of bacteria that no need to enter the cells. They colonize the mucous membranes or tissues and multiply there. Sometimes they stay at the biting site or at the site of entry. Sometimes they spread through the bloodstream and disseminate in the body. But then they stay outside the cells. So that's what we typically call extracellular bacteria. Then truly we may have intracellular bacteria, even obligate intracellular bacteria. They are very rare. The examples are just Rickettsia and Chlamydia. These are two types of obligate intracellular bacteria. So, but then mostly the pathogenic bacteria may be considered as facultative intracellular parasites. What does it mean? It means that they don't need the cells to multiply, the living cells. They have all their own biosynthetic machinery and enzymes and protein synthesis machinery to live independently on the cell, not inside the cell. But if they will be engulfed by macrophages, so then they can survive in macrophages due to the long list of virulence factors expressed by these bacteria. So altogether we may have situations with extracellular bacteria and intracellular bacteria. So let's start with the simplest way. This is the extracellular bacteria. Those that multiply outside the host cell. They can multiply in blood plasma, in the lumen of the gut, in the lumen of the airways, in the urinary tract, and etc. So, and various species of extracellular bacteria may cause diseases, and they cause diseases by two basic mechanisms. The first mechanism, the microbes colonize the surfaces and multiply there and induce inflammation that causes tissue damage, destruction at the site of infection. And then the second way how extracellular bacteria may cause diseases is bacteria may produce toxins, and these toxins may enter the bloodstream, and the toxins may enter the cells, and these toxins will induce diverse pathological effects. And among the toxins, we distinguish, and don't forget about it, endotoxins, that I attributed to the gram-negative bacteria lipopolysaccharide. So that is a very potent activator of the dendritic cells, the macrophages, and inflammatory response. And we also may have exotoxins that are usually secreted from the microbe, by the microbe into the environment. So, and they have variety of mechanisms. So sometimes they may just bind with the cell and kill it, cause it is osmotic lysis. So other toxins may interfere with the normal cellular functions and metabolic activities, protein synthesis, or the electrolyte balance and so on. So, and also toxins may act as super antigens, right? So, and these super antigens can stimulate the production of cytokines that cause damage of the host tissues. By the way, okay, we are talking about bacteria yet, right? So then, what are the clue mechanisms that protects us against extracellular bacteria? That some people wrote in immunology classes about this. They wrote that complement activation, phagocytosis, and inflammatory reaction are the most important mechanisms of innate immunity to extracellular bacteria. Yeah, that's good, but not enough. So, first stage or first important thing is activation of phagocytes and inflammation. So how the activation of phagocytes occur? My dears, this is a good time to repeat the surface structure that occur on the surface of macrophages. So, and that is a very small example of it. Can you see here? These are different types of molecules that may bind with extracellular bacteria or fungi or viruses also. So then could you see please that different types of receptors may communicate with different types of the pathogen. And see here, we may have receptors that bind with the viruses, receptors that bind with bacteria, Minkel receptors, receptors, dectin-1, that bind with fungi and bacteria, the receptors that may bind with the bacteria and the helminth eggs. So we have variety of those receptors that are involved in binding and recognition over the pathogen-associated molecular patterns that occur on the surface of the microbe. Regarding some more receptors of the macrophages, please take a look. The very important one is mannose receptor, but it may, by the way, recognize bacteria and fungi very well because they recognize certain terminal sugars on the surface of the carbohydrates of bacteria and also fungi, right? Then we have, you may not know mannose, lactose, and so on, but you know the structure of the bacterial cell wall. Then we have a lot of so-called scavenger receptors. And here you have example of the scavenger receptors A or CD36 of the macrophages. And they recognize bacteria and mediate the engulfment of lipopolysaccharide, some lipoproteins, lipoteichoic acids. Please, I would like to underline this because this is a very important pump for gram-positive bacteria. Nucleic acids, beta-glucans that are found on the fungal cells. So then the binding with these receptors mediate the phagocytosis of the microbes. Then on the surface of macrophage we may also have so-called FC fragment receptor, FC gamma receptor, for example. So, and this is a receptor that binds with the FC portion of the immunoglobulin when the antigen-antibody complex is formed. So it means that is activation through this receptor is the result of so-called opsonization. And the macrophage usually has the CD receptor that recognizes complement molecules. So the subcomponents of the complement system also recognized as the opsonins, C3b or C3d molecules, by the way, right? So, and it is well known that macrophages binds much better with the microbe through self molecules like CD3 receptors or immunoglobulins, then better than through so-called TLR receptors. So these TLR receptors and another group of the receptors that can recognize the microbes, and we mentioned them. So the most important for bacteria 1, 2, 4, 5, 6, 9, 11 that mostly stimulate the phagocytic activity. Then we have mycobacteria and corynebacteria. And you know these two guys are a bit different from the others because they are covered with wide range of glycolipids that can activate macrophages through different signals, and this signal is given here, right? Special for mycobacteria and fungi, by the way, also. So, and these include glucose and trigallose mycolates, and they are closely related corynομycolates that are found in corynebacteria. Okay, we have discussed it, and now we see that how innate immunity will be activated against extracellular bacteria. You can see first the activation of complement system through mannose binding protein, right? So, or alternative pathway. Do you remember that these are the pathways that will be activated first by unfamiliar to the body bacteria, or by the way fungi. So they bind also with the mannose receptor, CD14, TLR group of receptors. Dectin receptors are involved mainly in fungi uh, uh, binding binding with fungi. And also you can see bacteria that may have the special injection system, and they go through the membrane. So once it happens, the signaling, can you see? These are signaling mechanisms. Do you remember? From adapter molecule, the information is sent to the nucleic factor, for example, an FKB factor, and these regulate the bind with the DNA genome and regulate the expression of new proteins in the nucleus. And also you can see here that this is phagolysosome or phagosome or endosome. We also may have the specific TLR receptors, and the hooks will be directed inside the endosome because they recognize the unusual motifs of genotype of bacteria or unusual repeats in the genomes of the viruses, right? So then, in turn, they also perform signaling to the nucleus to activate the production of new proteins to cause inflammatory response. By the way, it is shown here. The result of all those activations will be first production of interleukin 1, 6, 8. So, and here you can see this result, right? So you've seen this picture, how it happens through the adapter molecules. 3 for my D88. These are protein kinases that activate an NF-kappa factor, change the expression of the genetic material, and different cytokines and interleukins can be produced, co-stimulatory molecules for antigen presentation occurs. So when we have a stage of acute inflammatory response. And as a result of innate immunity against extracellular bacteria, the activation of phagocytes causes the phagocytosis of bacteria and secreting of the cytokines. And these cytokines are here, right? And they induce very strong inflammatory immune response with leukocyte infiltration into the site of infection. And variety of the infections caused by Staphylococcus, Streptococcus, Neisseria, and others. So, cause this type of the inflammation, and they recruit leukocytes that engulf and destroy bacteria. But most probably, as you know, for innate immunity mechanisms, the result is the control of multiplication, but not elimination of the microbe. So for elimination of extracellular bacteria, the humoral acquired immunity gives us the major best protection against extracellular bacteria, and don't forget, against their toxins, because a lot of bacteria produce toxins. If you take a look at, these are bacteria, right? And they secrete the soluble substances that are called exotoxins. And mainly, if and when antibodies are produced by plasma cells and released into the environment, those antibodies may play a critical role in the elimination of these bacteria. So they cause opsonization, and opsonized bacteria may cause the stronger digestion of the macrophages, and also they may perform neutralization of the toxins and many other effects. So, and uh, the cytokines that will be released, that we mentioned already, and some others. So the cytokines of the helper cells, interleukin 17, 22, and tumor necrosis factor. They activate macrophages and neutrophils to increase the inflammatory response. And the extracellular bacteria induce mainly Th1, Th17, and also Th2 immune responses. So, the result of inflammation, they cause the activation over the macrophages and neutrophils, cause tissue damage by local production of the free radicals and lysosomal enzymes. Example of it can be the development of the rash, because that is the sign of the uh, tissue damage. And also secreting of the cytokines like this, right, stimulate the synthesis of acute phase protein, C-reactive protein. They cause systemic clinical manifestation of the infection. But usually the inflammatory reaction in those reactions is controlled and self-limited. So, few words about the bacterial toxins. You can see here that some toxins may act as superantigens. And superantigens, they may bind with, in between on the outside, in between the immunological symptoms, synapse development between T cell and antigen presenting cell when they bind together to present the antigen to T cells. And then superantigens bind from outside and cause the very strong activation of both T cells and antigen presenting cells. So the result of this, the example of these antigens are TSST of staphylococci, enterotoxin of staphylococci, then pyrogenic toxin of Streptococcus pyogenes, and they cause the interleukin storm. So, and the cytokines produced within this storm can cause systemic inflammatory syndrome. So, and also the antibodies produced against particular types of bacteria may produce the tissue damage as a result of the immune response development. And for this, the most famous are streptococci that may cause disease-generating antibodies, synthesis of disease-generated antibodies. So, you know the example. The first example is rheumatic fever. So, and this is caused by the cell wall M protein that have cross-reactivity with the myocardial proteins. And these antibodies will be deposited in the heart and subsequently cause carditis. The second sequel of the streptococcal infections can be post-streptococcal glomerulonephritis, when antibodies produced against the microbe form complexes with the bacterial antigens, which are deposited in kidney and cause nephritis. So, and here we have our view of the humoral acquired immune response that provides a major protection against extracellular bacteria and their toxins. First, when bacteria will be binding and activating B cells, and B cells also may act as antigen presenting cell for T cells for protein antigens. So the B cells will be activated and differentiate. So B memory cells will be released by also antibodies that are released from the plasma cells. What they can do? First, they can bind and neutralize bacteria. Then second, they may cause opsonization through binding with Fc gamma receptor, and the phagocytes will engulf them much better. And also while they activate the complement system, then they cause opsonization, it causes opsonization with C3b molecule. So we have phagocytosis of C3b coated bacteria and phagocytosis of IgG coated bacteria. Then the complement activation causes the release of C3b for opsonization, the involvement of neutrophils in the process and improve inflammation, and the lysis of the microbes. So then the dendritic cells activation by bacteria and acting of them as antigen presenting cells, they may also cause the differentiation of T helper cells into interleukin 17 cells. So, and the cytokines produced by these cells, for example, interleukin 17 and 23, the strong activator of the neutrophils, interferon gamma, the strong activator of macrophages, and different cytokines, the strong activators of the B cells to produce antibodies. That is the summary of how antibodies, or summary of how antibodies participate in protection against extracellular bacteria and their toxins. Now take a look at the facultative intracellular bacteria, or generally intracellular parasites. So they survive within phagocytes. And the result of it is that they are inaccessible to circulating antibodies. So, my dears, antibodies cannot bind directly with them. Intracellular location protects them from being killed or being affected by antibodies. So then the elimination of this type of bacteria will be the function of cell-mediated immunity. And the antibody immunity may just cause some additional effect, or no effect, like for example mycobacteria infection. Antibodies will be produced, but they will be not protected. So, and in many infections caused by intracellular bacteria, the host immune response causes tissue damage, and mycobacteria is the best example of it. So, what is the best way to remove the intracellular bacteria? Again, this is phagocytosis and natural killer cells. That's how we show natural killer cells. See? Born to kill. They are this. So, but these two, this neutrophil and this NK cell, they act differently. So, phagocytes and neutrophils ingest and attempt to destroy intracellular bacteria. So, initially neutrophils act upon intracellular bacteria, and later macrophages will be involved in the process. So then you should ask, what is the activity of NK cell? They can kill the host cell infected by intracellular bacteria, not by ingestion, but by releasing some lytic molecules that may kill the host cell together with the bacteria inside. And what is important, that this can be arranged before the antigen-specific T cytotoxic lymphocytes will be activated. So it means more rapidly than T cytotoxic lymphocytes. That is a very important role of the innate immunity mechanisms in eliminating of intracellular bacteria. So, additional activity of the NK cell, that it may produce interferon gamma, the strong activator of macrophages. And again, the best uh, uh, producer of gamma interferon is the Th1 cells, but before they are activated to release interferon gamma, the NK cells can do this to activate macrophages. So, and I just want to repeat that NK cells may kill microbe-infected cell. This is called infected or autologous cell. It starts to be autologous by different ways. And here we can see the way that usually happens when bacteria infect the cell. So see, MHC class 1, is still there. So it will be not produced in a cell if virus is infected. So when bacteria infect the cell, so usually they have own synthetic mechanisms. They don't really take over the biosynthetic mechanism of the host cell. So that's why MHC1 is still produced on the surface of the cell. So the target cell expresses the ligand for inhibitory receptor. See, there is the inhibitory receptor. But then usually during the infection of the cell with the intracellular bacteria, a lot of ligands for activating receptors will be produced. So they will give overwhelming, very strong signal for the multiple activating receptors of the NK cells. So a lot of protein tyrosine kinases are produced, and the protein tyrosine phosphatase can inactivate one but not all signals produced by multiple ligands for for activating receptors. So the NK cell will be activated. So and produce the very strong signal perforin and enzymes to kill the intracellular bacteria infected cell. So adaptive immunity to intracellular bacteria mainly is mediated by T cells. And this is a familiar picture when bacterium is engulfed into the dendritic cell, right? And CD4 cell recruit and activate phagocytes by producing ligand CD44 additional signal with the binding with the dendritic cell of macrophages. And then the secretion of interferon gamma cause macrophage and dendritic cells activation. And then naive CD4 T cells will differentiate into Th1 effector cells to produce interleukin-2 and gamma interferon, so the naive cytotoxic T lymphocytes stimulated by phagocytosed bacteria can kill infected cells directly. So they go and kill infected cells. So, okay. Oh, sorry. So, in this case, CD4 T cells will cooperate. This is a CD4, and these are CD8 will cooperate with CD8 positive T cells, and they may arrange the defense mechanisms against such microbes as Mycobacteria to tuberculosis at less extent by against, for example, Listeria monocytogenes and other intracellular bacteria. So, because they can survive in phagocytes, so by escaping into the cytoplasm, and then they may persist in macrophages. So, and the activated Th1 cells will be secreting gamma interferon and tumor necrosis factor that stimulates macrophages. So, and then macrophages will perform the oxygenic burst to kill the bacteria inside, or to commit a suicide and kill bacteria inside. Next pathogen that we discussed today is the virus. And before we discuss the immunity against viruses, I would like to to um, revise what properties of the viruses will be very important in organization of the immune response against intracellular parasites. Very specific intracellular parasites, different from intracellular or facultative intracellular bacteria. So, and for the first, what is the most important feature is that viruses are obligate intracellular parasites. And that's because they multiply or replicate in the host cell only, because they don't have their own biosynthetic machinery. They take over the host cell machinery, the human cell machinery. So, and the viruses have a unique way of replication. When one virus enters the cell, and thousands copies can be released from the infected cell. So, and the components of the virions are produced in different compartments of the cell. This is called disjunctive way of replication. So, the components will be produced in different compartments, and then they will self-assemble together, and then the new viruses or virions will be released from the host cell. And that is the moment, and that is the process that really important in understanding of the pathogenesis of viral infection. Because first, viral replication interferes with the normal cellular protein synthesis and function that may lead to the death of the cell. And also the infected cell may die usually during the release of newly synthesized virions. So those viruses are called cytolytic, that kill the cell during the release of the newly synthesized virions. And some viruses, and for viruses that's really typical for a lot of viruses, they may cause so-called latent infection, so they escape from the immunity. And when we are talking about the immunity against viruses, innate and adaptive immune response are aimed at the blocking infection and eliminating virus-infected cells. I think this is it, right? Virus-infected cells. So the virus is protected by intracellular location. So that's why the whole cell should be killed. So, and several interferons will be produced when the cell is infected with the virus, right? And interferon alpha produced by virus infected cell activate NK cell that may eliminate the virus infected cell as a part of innate immunity mechanism. Then the neutralizing antibodies are important in fighting with some viruses that have the extracellular stage, and these are usually uh, non-enveloped viruses like enteroviruses, for which the development of the neutralizing antibodies will be enough to fight with infection. But also for most enveloped viruses, T cytotoxic lymphocytes are needed for eliminating virus-infected cells from the body, and they make the major mechanism of adaptive antiviral immunity. So, and here you can see the principal mechanism over the immunity against viruses. These are NK cells. And the inhibition of the virus replication by interferon mechanisms. And I hope you remember how interferon works. It protects, it causes the antiviral state development in the neighboring cell, cell neighboring to the, with the virus infected cell. Okay. So you can see here how viral pumps, such as nucleic acids, right? See, nucleic acids may be recognized by the endosomal TLR receptors, TLR3 and 7 to 9. And these receptors activate the transcriptional factors. They are shown here. And those transcriptional factors bind with the genome of the cell and change the expression mainly of the proteins that are interferons. So NK cells, and you can see here, right here, that quite a lot of cells infected with the virus, because the viruses shut down the cell protein synthesis and switch it to the viral protein synthesis. The MHC1 will be not produced. This is called missing self recognition, right? So the NK cell could not recognize the normal cell as normal because of the missing signal MHC1 molecule. So then MHC1 molecule is missing. No inhibitory, there's no ligand for inhibitory receptor, no inactivation of protein tyrosine kinase that result from the activating receptor and the ligand for this receptor interaction. So then NK cell will be fully activated and the granules will, with perforins and granzymes, move towards the site of contact with the target cell, and then they will be released to kill the virus infected cell. So generally, that were the innate immunity mechanisms, and these are the acquired immunity mechanisms. Against some viruses, both antibody-mediated and cell-mediated immunity are equally important. Why? How neutralizing antibodies work? Because when they bind with the virus, for example here, so you can see that virus binds with the receptor and through it enters the cell by endocytosis. Then if the antibodies block the surface proteins of the virus. Then the virus will fail to bind with its receptor on the surface of the cell, so the reaction will be stopped at the very initial stage. This is called virus neutralization. So the antibodies in our body may neutralize the viruses and make the infection abortive. Uh, but also, of course, the most important um, mechanism of virus elimination is the T cytotoxic cell activation and killing of the virus infected cells. And the elimination of the viruses mainly uh, connected with the activation of T cytotoxic lymphocytes. Because we may have a sort of viruses like paramyxoviruses, automyxoviruses, HIV viruses. So they usually reside inside the host cells. So then, uh, the elimination of those cells depend mainly on the T cytotoxic lymphocytes that recognize the presence or the absence of the molecules on the surface or presence of the MHC1 with the viral antigen on the surface of the virus infected cell. And again, the TCR receptor recognize the antigen and CD8 receptor recognize the MHC1. Then the activation of apoptosis occur and the cell will be eliminated. Virus infected cell will be eliminated from the body. So the full differentiation of the CD8 positive lymphocytes is needed to produce the fully differentiated cells that may be formed, produce of the gamma interferon and the production of the perforins and granzymes to kill the virus infected cells. And this is a process of virus elimination from the body. So then we have to take a look at the latent infections. So it is very common for DNA viruses that may persist in the cell or in the body for a very long while. Quite often, viruses in this stage are not multiplying. So that's why if the viruses are not multiplying, the viral antigens are not produced and released into the body, so the immune response will not be developed against these viruses. So, and the immune response that will exist may control the multiplication of the virus, but usually cannot eradicate the virus from the body. So, and when with ages or under some conditions the immunity drops, then the viruses will get chance to multiply again, and they may multiply and cause cytopathic effect and release again to the body and spread in the body. So the example are herpes viruses, and one of those is Epstein-Barr virus, or for example, the virus from another family, hepatitis B virus. So these are RNA viruses that are capable of causing latent infection, so they usually overcome immune defense mechanisms. So, how viruses may evade the immunity. So Antigenic variations of many types of the viruses may do this when the antibodies produced against previous type of influenza or HIV or rhinovirus that exist in 100 serotypes. So the antibodies against one serotype will not neutralize the microbe from another serotype. That's one way. Then inhibition of antigen processing. So a lot of viruses may produce the substances that block the transporter associated with antigen processing. So they stop the processing of the viruses. So, and these are herpes and cytomegalovirus. Then some viruses may produce usually these are non-structural proteins that are analogs of cytokine receptors. And this is typical for vaccinia, cytomegalo, and pox viruses. So then, they also may produce some immunosuppressive molecules or cytokines. It is known that Epstein-Barr infection may cause cells to produce interleukin-10 that downregulate the cell-mediated immune response. So the only antibody immune response will be developed that will be not highly protective. So it may control the multiplication of Epstein-Barr virus, but cannot eliminate it from the body. And also the viruses may infect and cause death over the immune cells. For example, HIV may cause death of the CD4 lymphocytes, and Epstein-Barr virus may cause death of the B cells. So, of course, it will impair the ability of the immunity to eliminate the viruses. Now we take a look at another group of pathogens that are less familiar to you, and these are protozoa. These are protozoa. Then fungi, you see two candida inside the cell, inside of the human cell, and helminths. So we will discuss it briefly because this is, you know already a lot of things, but then you need to know the most important features of these immunities. So first, immunity to fungi. First, we need to confess that mostly the fungal infection occurs in immunocompromised hosts or during the transitory immunocompromission of the host. That's first thing. Then the second thing to understand. Different types of fungi may occur extracellularly or intracellularly. For example, you can see Histoplasma capsulatum, that is dimorphic fungus. In the soil, it forms the molds, but as soon as it enters the body with the 37 degrees Celsius and ingested, so it turns into the yeast-like structures. So when you can see the intracellular location of this Histoplasma capsulatum. And the immune reaction to fungal infection are always based on the combination of the responses to extracellular and intracellular parasitic organisms. So what is important in innate immunity to fungi? The basic protective mechanisms are activation of neutrophils and macrophages. And those cells, they have very special TLR and Dectin 1,2 receptors, so to bind with the fungal cells. So you can see here the dendritic cell decorated with antifungal pattern recognizing receptors. So, and upon recognition of fungi through this receptor, the signaling mechanisms through already well-known and new adapter molecules, for example, with me D88 and others, right? So, and the signaling mechanism should be uh, uh, transferred from the or transduced from the surface into the nucleus, and it will lead the generation of Th1 and Th17 adaptive immune responses and activation of innate effector cells. So, neutrophils and macrophages release fungicidal reactogenic substances and lysosomal enzymes. So, and neutrophils and macrophages engulf fungi and try to kill them intracellularly, and the activity can be inhibited with some fungi. For example, there are strains, so that may inhibit the production of tumor necrosis factor and interleukin 12 and activate synthesis of interleukin 10. And by the way, within those types of infections, if we take a look at the spectrum of interleukins in the body, if we have interleukin 10 produced high level, so that will be poor prognostic sign of the infection. So, and if we have high level of gamma interferon and interleukin 12, it will be very good prognostic sign of the infection. So now a few words about Candida. So Candida may occur in two different types in the body. So they may occur as yeast-like structures or as the pseudohyphae. You can see here the yeast, the Candida organism that forms already the germ tube and pseudohyphae. But then this pseudohyphae is too large to be ingested by one single neutrophil. So, and neutrophil tried to induce highly fungicidal substances and at least to turn it into pieces, right? And these are the substances produced within the oxygen-dependent pathway, and the activation of them will depend on tumor necrosis factor and interferon gamma. So, and if we have there's a very nice electron photograph, there's a very nice picture, and you see here that how the Candida form the pseudohyphae and pseudofilamentation, and then neutrophils may collect together and form so-called intracellular fibers. So neutrophil extracellular traps. So you can see those traps, and then they trap the Candida there, and the neutrophil extracellular traps contain neutrophil elastase, and on the special immunofluorescent assay it turns green, right? And then you can see DNA is blue, and Candida albicans is stained red here. A hypha is partially engulfed by neutrophil and additionally trapped by extracellular traps, and that means that neutrophils may occur extracellular and intracellular digestion simultaneously. The complement can be can make very good protection against fungi because it can be easily active. The complement system can be easily activated through the multiple both three pathways. So through the lectin pathway, because it has a lot of mannose residues on its surface, fungal cells, then they can be activated by alternative pathway, and they also can be activated by classical if antibodies are already there. So it may result in the membrane attack complex formation. But unfortunately, fungi may avoid the complement-dependent killing. Why? Because they may produce complement inhibitors that interfere with the cascade activation. Then the fungi, they are much more complex microbes to compare with bacteria or viruses. So they may produce a variety of the hydrolytic enzymes and proteases that may degrade the complement system components. So while they degraded, the membrane attack complex may not be produced. And also that's important, opsonizing molecules like C3b will not be produced. And the mediators of inflammation will not be produced. So they may evade the activity of the complement system. And by the way, if we take a look at the fungal cell, it consists of not only glucans, but it has a hard layer of the chitin. So the fungal cell wall is really very thick, and the formation of the membrane attack complex on this surface may be not maybe not enough for subsequent lysis of the cell. So that's how the fungi may escape activation of the complement system. So the fungi may activate very well the dendritic cells because they have very special dectin 1 and 2 receptors. These are lectin-like receptors that bind with the fungi, and as a result the dendritic cells will be activated to produce very strong pro-inflammatory signals, interleukins 1, 6 and 23, that activate the naive T cells and also neutrophils. So these cytokines cause or stimulate the inflammatory immune response and cause differentiation of the naive T cell into the Th17 cells. So we think that Th17 cells are very important in protection against fungi. So they have the ability to induce the inflammatory response and cause the barrier function. So those signals produced by Th17, and except for interleukin 17, they are IL-21, 22, 25, and tumor necrosis factor. They stimulate the inflammatory reaction here, and by this they participate in the clearance of extracellular bacteria, fungi in gastrointestinal tract, because this Th17 mainly occurs in the mucous membranes. That's we did. And here I would like to demonstrate that we just touch a little bit the antifungal immune response. If you take a picture of this slide and see it in the hostel, so you can see that it is much more complex. And here we can see the protection against candida yeasts that are induced by phagocytosis and engulfed into the resident macrophages. They start inducing interleukin 1 and tumor necrosis factor. The neutrophils will be stimulated to come to this area and produce highly reactogenic oxygen free substances that are called free radicals to kill the fungi and to produce the extracellular neutrophil traps when the formation of pseudohyphae occurs. So, and then we also should mention the NK cells. They come and produce perforins and granzymes to kill the candida cells. And also we have to take into consideration that dendritic cells cause the T cells differentiation into Th17 cells that can produce very important substances, defensins and also interleukin 22 that will be exposed on the surface of the mucous membranes and have very strong antifungal uh, activity to destroy candida hyphae or candida uh, or candida cell over the unicellular organisms on the surface of the mucous membranes of the epithelial cells. Okay, then we go for summary of antifungal immunity. So, innate and acquired cell-mediated immunity is the most important mechanism. Neutrophils or macrophages act from behalf of innate immunity, and extracellular fungi may generate strong CD4 Th17 immune response through the activation of dendritic cells through the Dactin-1 receptors. And dendritic cell will cause the differentiation of T cells into Th17 cells to produce interleukin 6, 23, and 17. They stimulate the inflammatory response and activate neutrophils and macrophages to destroy fungi. Some fungi that occur intracellularly, like Histoplasma capsulatum, may need T cytotoxic cells activation and killing of the fungal infected cells. Antibodies can be important also. For example, if IgA antibodies are produced, they may prevent binding of the fungi with the mucous membranes. IgG may act as opsonizing molecules, and AgE that are under Th2 activation completely may serve as a prognostic sign of fungal infection due to the inhibiting of antifungal immune response by Th2 cells. So now a few words about protozoa, very briefly. So first, the importance or the features of protozoa important for development of the immunity against these microbes. And this table is given just to make your remember or to just revise what kind of protozoa do we have. So, and these are parasites, protozoal parasites, that usually cause not acute but mainly long-term chronic infections. So then the major virulence factors of protozoa are the variety of hydrolytic enzymes that may cause the lysis and the damage of the host tissues, and the activation of the hypersensitivity reactions. But also what is important in protozoa from the point of view of the immunity. So they may change the forms or when they stay in the body. For example, they may form vegetative forms and cysts. And these two different types of forms may have different antigenic structure. And you know that shift in antigenic structure may interfere with the effectiveness of antibody immune response against protozoa. And we may have extracellular and intracellular protozoa in the host. And here you see a very nice picture. When you see a malaria protozoa have multiplied into cells in a culture dish where we have red blood cells. So here they multiply. And one has burst already, opened, releasing the parasites to infect other cells. So they are intracellular parasites. And the immune response against protozoa limit pathogen multiplication and rather prevent reinvasion than eliminate the organism. It means this immunity against protozoa is usually called non-sterile immunity. Why non-sterile? So the immune mechanism will cause or will inhibit multiplication and dissemination of the microbe, but cannot eliminate the microbe. So many protozoa organisms may survive and replicate within the macrophages and so here the leishmania is given as an example that may reside in some phagocytes, and it even may multiply in the phagocytes and many other different types of cells. So these pathogens may generate very weakened innate immune response and evade attack of acquired immunity. So, and you can see here that regarding the pumps on the surface of parasites, they have also the pathogen-associated molecular patterns that usually may bind with some TLR receptors. For example, TLR2 and TLR4 can be activated by phosphatidylinositol lipids that happens on the surface of the malaria organism, for example, Plasmodium species, or that may happen on the surface of Toxoplasma gondii and other Toxoplasma, and also Cryptosporidiosis, but this is the fungus already. So protective mechanisms of immunity are similar to those that may help to eliminate viruses and intracellular bacteria. Macrophage activation by cytokines from Th1 cells, and this is major one is interferon gamma, may be important in clearing of the infection by such as malaria, cryptosporidiosis, and toxoplasmosis. T cytotoxic lymphocytes response is needed also to eliminate plasmodium of the malaria. And this is important defense mechanisms against malaria. Antibodies immune response also may be important as opsonizing molecules on the surface of the protozoa. So, but then the different protozoa regarding their structure, chemical composition, and the type of parasitism may evoke different types of adaptive immune responses. For example, for Plasmodium species, the basic immune mechanism is killing with the help of T cytotoxic lymphocytes and antibodies. For Leishmania donovani, mainly the macrophage activation by Th1 DH cells. This is type 4 hypersensitivity reaction is important. And for example, for Entamoeba histolytica, the causative agent of amoebiasis, the phagocytes and antibodies are the most important mechanism of elimination. In the again, you can see the picture that quite a lot of protozoa may have some tricks to survive within the macrophages. So the cell-mediated acquired immunity to protozoa depend on activation of specific to Leishmania Th1 cells and release of gamma interferon that activates macrophages. But then it is important that, for example, mm, some of Leishmania or some of protozoa like Leishmania may produce macrophage suppressive cytokines like interleukin 4. So Leishmania activate Th2 cells to produce cytokines that will downregulate the cell-mediated immune response. This is linked with the progression of this disease. Okay, so now a few words about helminths because just 10 minutes left. So let's take a look at the helminth parasites from the point of view of immunity. Let's take a look at their unique structures. So you know that helminths are parasitic worms that feed on a living host to gain nourishment and protection. So they cause poor nutrient absorption, weakness, and the disease in the host. The worms and their larvae live in the bowel and are referred to as intestinal parasites, many of them. They are multicellular, very large organisms that are visible to the naked eye. For example, this is the Schistosoma mansoni, that is the parasite of a trematode class, that is the agent of the disease, quite common, that is schistosomiasis. Okay, so then, immunity to helminthic parasites. So, in contrast to protozoa, helminths may survive in extracellular tissues. They don't need to enter the cell. They are extracellular parasites. So that's why the elimination of these worms often depend on the antibody immune response. It is very important in the immune response. And then, okay, maybe it's a bit difficult for you. Take a look here. So let's take, what is the innate immunity to the worms? Phagocytes participate in immune response to helminths, but usually they are too large to be ingested, right? They are up to 140 micrometers in length. So, and when phagocytes produce microbicidal substances, the worms may resist these substances. It means that macrophages and neutrophils are not so effective against worms. So we have to take a look at the eosinophils because worms may resist macrophage and neutrophils cytocidal substances. Why? Because the worms are covered with so-called tegument. What is tegument? It is outer body covering of helminths that consist of, these are biopolymeric. It consists of proteins, lipids, carbohydrates, and even RNA. So the tegument, thick tegument is a very good protective layer for the worms. And it has also not only protective but secretory and absorptive function. So, but then it is very important that these glycocalyx responsible for inhibition of host digestive system that may cause malnutrition in the host. And the acidic glycosaminoglycans of the tegument inhibit the number of digestive enzymes of the host. And how it is linked to the immunity. So the complement activation may occur mainly through alternative pathway in the beginning. So but then some parasites may develop resistance to complement-mediated lysis due to the very strong covering of the body, and also the uh, by the production of the enzyme that may degrade the complement. But then generally, we have a step in the complement activation when C5a molecules are released. Then those C5a molecules may be very important because they are chemoattractants not only for neutrophils and macrophages, but also for eosinophils. And we come to the eosinophils because they are very important protective mechanisms against the worms. Eosinophil activation may cause also the production of immunoglobulins E, and these are the principal protective mechanism of the immunity against many infections caused by parasitic worms. So this is mediated by Th2 cells. And parasitic worms stimulate differentiation of naive CD4 cells into Th2 effector cells. They start producing interleukins like interleukin 4 and 5. And these are activators also and stimulators of the production of the class switching from IgM to IgE. And these antibodies bind with Fc epsilon receptor on the surface of the eosinophils and mast cell molecule and mast cells. And this is activate the maturation of eosinophils and the other granulocyte cells. And the eosinophils may produce the substances so that are in their granules that are called basic proteins. And then the eosinophils accumulate in inflammatory infiltrates at the site and causes the hypersensitivity reaction, and then the eosinophil the granulation causes release of a lot of very important anti-helminth substances. These are eosinophil peroxidase and toxic proteins like lysosomal hydrolases, major basic proteins, and eosinophil cationic proteins. These substances have very strong cytocidal activity for the worms and bacteria, so as well as for the human tissues. So eosinophil peroxidase is distinct from that of the neutrophil, and it promotes synthesis of hypochlorous and hypobromous acid, which is destructive to parasitic worms, protozoa, and sometimes human cells. This is the major way. And eosinophil have surface Fc receptors for immunoglobulin G, A, and E antibodies, so multiple Fc, gamma, alpha, and eta receptors. An antigen binding to the receptors associate with cross-linkage of these receptors, and it causes very strong degranulation of the eosinophils. That's how antibodies may improve eosinophil activity against the helminths. So they will release the cytokines and mediators like prostaglandins, leukotrienes, and some other substances that cause very strong allergic inflammatory reaction in the site of their activation. So they cause so-called barrier immunity, and this is expulsion of the parasite from the intestine. And it depends on the combination of eosinophils and mast cells that may release histamine. The expulsion of some intestinal nematodes may not require immunoglobulins E, but happens with eosinophil activation, eosinophil activate degranulation directly. And by the way, so interleukin-4 dependent activation of intestinal peristalsis also may participate in the elimination of the worms from the gut. I think with this we have to finish, and we come in time exactly. So with this I would like to finish my lecture, and my dears, I hope it will help you next week to answer the antiviral, antiprotozoal, and anti-helminth immunity. Take one more look at the lecture within several hours. Take pictures of it so it will help you to answer the questions. So with this, my dears, I would like to say goodbye to all of you. And next week will be microbiome week when we go ahead with some more viruses. Goodbye, my dears.